Hey everybody, jumping right into Chapter 6, Introduction to Pharmacogenomics. We're going to discuss the genetic differences in drug metabolism as well as the clinical implications of pharmacogenomics. Our overview. All right, so Introduction to Pharmacogenetics and Genomics. This is often referred to as PG with the little x, is a branch of science that's concerned with the identification of the genetic attributes of each individual that leads to the variable responses to our drugs. So in other words, what makes me different than you and how I'm going to metabolize the drug over you? The science of DNA, which is deoxyribonucleic acid, so the science of DNA analysis for the purpose of identifying gene variations that impact medication responses. Until recently, the ultimate goal of pharmacogenomics has been the development of the prediction models to forecast debilitating adverse events in specific individuals. But more recently, across populations based on the similarities in age and gender, or like race or ethnicity, we can find differences as well. Okay. Genetic differences in drug metabolism and the disposition of drugs will contribute to the variability in our drug responses. Genetic knowledge then helps foster greater personalization of pharmacological care. The genome, the genetic material of an organism. So all of the DNA contained in any individual cell is called the genome of the individual, right? So it represents the genes that an individual can express. Interestingly, even though two unrelated people share about 99.9% of the same DNA sequences, with less than the 0.1% difference between them translates into differences in 3 million nucleotides. So these variants introduced that we've talked about are the SNPs. And the SNPs are the single nucleotide polymorphisms, which is the variation of a single base pair in a DNA sequence that accounts for individual genetic differences in drug metabolism. Reading through the chapter, and it talks about the emergence of mapping the human genome was a huge undertaking and finished in the early 2000s and really has broken science completely wide open. We know that about 95% of the sequence of all human DNA was established during that time, and that has resulted in the identification of the open reading frames for many human proteins. About 1.4 million SNPs, or the SNPs, in humans have been identified through a mass effort by the SNP Consortium, or the SNP Consortium. It's a collaboration among 10 pharmaceutical companies, the Wellcome Trust, which is a research charity, and 5 academic centers. Box 6.1 in your book is a nice quick resource as well for some of these definitions of the things we've been throwing around out here. Like I said, looking back to the history of how we've gotten to here is really neat. So I do think it's nice to go through. 510 BCE, first inter-individual differences of a drug administration noted with hemolytic anemia in the 1950s. 1959, pharmacogenetics is first coined. In the 1950s, they identified hemolysis in Black men. In the 1970s, they realized that there's slow acetylators or slow metabolizers in certain ethnic groups. So we are going to visit about the genetic differences in drug metabolism. So we have genetic polymorphism, phase 1 and phase 2 metabolism we'll talk about, and then again the CYP450 enzymes. So when we recognize that the patient's DNA sequence might be used to enhance our drug therapy to maximize efficacy, we can begin to target drugs to patients that are likely to respond. It'll help us avoid adverse drug reactions and could potentially decrease the cost of healthcare in America. So studying the genetic differences is a very helpful tool. It helps us understand genetic polymorphism and how we can overcome that. So a genetic polymorphism occurs when a difference in the alleles responsible for the variation is a common occurrence. An allele is an alternative form of a gene. So a gene is called polymorphic when the allele variations occur throughout a given population at a stable rate of less than 1%. Under such circumstances, variant genes will exist somewhat frequently alongside wild-type genes. The variant genes will encode for the production of variant proteins in these populations. Monogenic traits by themselves cannot explain the complexity of drug metabolism. There are 4 phenotypes that affect genetic polymorphisms in individuals. Okay, so the first one we're going to talk about is poor metabolizers. Then we can have intermediate metabolizers, and then we could have extensive metabolizers and ultra rapid metabolizers. Okay, if someone has if they're a poor metabolizer, what are we saying? They don't have a working enzyme, right? They cannot metabolize the drug correctly. Intermediate metabolizers are heterogeneous for one working wild-type allele and one variant allele. Extensive metabolizers have 2 standard functioning alleles, but ultra-rapid metabolizers have more than one functioning copy of certain enzymes. Okay, in your book, Table 6.1 is a nice book to look at when we're talking about these things. It gets very busy, but it does kind of give us an illustration that we could look at. So if someone is a poor to intermediate metabolizer, what is that effect going to play on their drug metabolism? They're going to be slower at metabolizing the drug. So a prodrug will be metabolized slowly to the active drug metabolite. So this may have accumulation then of the prodrug, right? It's lingering longer in the system. The active drug metabolized slowly into inactive metabolite. So we could have the potential for accumulation of the active drug. So in other words, it makes sense to understand that if the drug's hanging around a lot longer in these people, then we oftentimes require a lower dose. Okay. If someone has extensive metabolism, we consider that to be the standard effect of drug metabolism, standard dosing, standard enzyme activity. It's kind of weird to think of it that way, that extensive is the normal. But ultra-rapid metabolizers have a really fast metabolism of the drug. So they're the opposite of the poor to intermediate. The prodrug is rapidly metabolized to the active drug with no dose adjustment. Active drug is then metabolized rapidly into the inactive metabolite. So these people need a higher dose of the active drug. Okay, so let's talk about the phases of metabolism in phase 1 and phase 2 metabolism, okay? In phase 1, we call this pre-conjugation. So drug metabolism, we know, generally takes place mostly in the liver, and it's divided into phase 1 and phase 2. Phase 1 metabolism is responsible for approximately 59% of the adverse drug reactions that's cited in the literature. Okay. In terms of evolution, the cytochrome CP450 or CYP450 enzyme system was one of the first biocatalytic machineries to emerge on Earth. What do we mean by that? So we started to look at the CYP450 enzyme system and understanding this phase 1 metabolism. During phase 1, we have oxidation, reduction, and hydrolysis reactions. In phase 2, we are now in the conjugated reactions, which is the attachment of the drug to polar molecules, making it highly water soluble with little to no pharmacologic activity. When we talk about the CYPs, the CYPs, they are generally located in the endoplasmic reticulum in the mitochondria in human cells. We find the CYPs mainly in the liver and the intestine, and to somewhat lesser extent in skin, brain, lung, and kidneys. Our hepatic, renal, and intestinal endoplasmic reticulum CYPs are involved in the biotransformation of a plethora of drugs and endogenous substrates in humans, mainly by oxygenation of the target substrate molecule and mediated by differential oxidation states of the central iron atom in the enzyme. All of that to be said is that because of this process, the CYPs are classified as monooxygenases. A lot of words there. Keep with me, this will make sense. When we talk about the CYP450 enzymes, okay, up to 19% of drugs are metabolized via the CYP2D6. So these CYP450 isoenzymes include the P2C9, P2DK, P2C19, etc. So the first enzyme we're talking about, specifically in your book, they start talking about the sub isoform or sub isoenzyme of the CYP450 is this CYP or CYP2D6. Okay. I like the way they break it down in your slides here. These are often involved with the beta blockers, antidepressants, antipsychotics, etc. here. They can have, this is why we can get tardive dyskinesia or narcotic side effects and the beta blocker effect itself. It's because of CYP2D6 genetic duplication in some ethnicities that causes them to be ultra rapid metabolizers. Okay. The non-functional 2D6 allele may increase the risk of the adverse drug reactions. It leads to variable therapeutic effects of things like tamoxifen. It alters the effect of codeine, and it's a really important different CYP450 enzyme. CYP2C9 involves warfarin. If you have a CYP2C9 2 or 3 polymorphism, you're going to need a lower dose because it's how you are going to metabolize warfarin. And it can be different by race. They talk about the different algorithms for the decision making, which were on your reading as well. CYP3A4 is a group of isoenzymes responsible for up to 50% of drug metabolism, right? So the CYP3A4 isoenzyme is responsible for metabolism of several important drug classes, that are used in primary care, like calcium channel blockers, antihistamines, or statin drugs, antiepileptics. Both drug-related induction and inhibition of our CYP450 specific enzyme 3A4 may complicate drug therapy in patients, so predicting the onset and offset of these effects is very difficult. So when people have this issue happening, the example the book gives is amiodarone, because amiodarone has a half-life of 60 days and requires months to reach a steady state and overcome the CYP450 enzyme system effectively. So if you have drugs that induce or inhibit the CYP3A4 enzyme, we have to monitor that closely. Which is why you have to follow up with people with medications in general, right? That's a very general statement to make, but it's true. Depending on whether or not people are poor metabolizers, ultra metabolizers, those things we're understanding through talking about the CYP450 system and all of these specific isoenzymes under it that you may see about in the literature. So you may look up a drug and you may see it's a CYP3A4 inhibitor. Therefore, you need to monitor this drug this way. The P-glycoprotein is a membrane-bound, ATP-dependent transport system, okay, that is responsible for the efflux of a variety of xenobiotics from cells to the extracellular fluid that plays a key role in the physiological defense system. So that is a lot of words to understand. To me, the easiest way to understand the P-glycoproteins is to think about it is that it is a biological barrier that will prevent many compounds from entering into the cells. So it's a transmembrane protein that then lines a barrier from drugs to be able to enter into the cell to work. Okay. So in order for the drug to cross the cell membrane, enter the cells to do its job, it can be limited if there is a P-glycoprotein issue. The general purpose of the P-glycoprotein is to protect the body from harmful substances because it would remove that's being absorbed in the intestines back into the gut lumen. It's an efflux pump expressed along the GI tract, and it limits the permeability of many drugs and affects their absorption and bioavailability. We have some drugs that purposely inhibit or activate both CYP450 and the P-glycoproteins at the same time. I'm going to use a YouTube video to help us further put together the CYP450 and the P-glycoprotein enzymes that maybe will make it easier to understand.
In this video, I'll provide a brief overview on the cytochrome P450 enzymes and discuss the possible drug interactions associated with it and what to do about it. Please make sure to like and subscribe to the channel. It costs $0 and it takes less than 5 seconds. Thank you. The cytochrome P450 enzymes. Now, these are enzymes that are normally found in the liver but can be present in many cells throughout the body. They play a huge role in metabolism of things that are foreign to the body. So in this case, drugs, and because of this function, is known for detoxification. Now, the original forms of drugs are usually not able to be cleared by the body, so the body must find a way to fix this. It does this by converting it into a more familiar form so that it can be excreted. It uses two methods. So you have phase one, which is the oxidation, or removing electrons from the compound so it becomes charged and more water soluble. And this is catalyzed by the CYP enzymes. Then you have phase two, which involves conjugation, which will further increase the compound's water solubility by attaching a group to it. So let's say we have a drug here and also the CYP enzyme here. Now try to visualize the drug or substrate binding to the CYP enzyme to form a complex. This would then release a more polar form of the drug. So drugs can affect the activity of the CYP enzymes. The effects can be characterized as strong, moderate, or weak. Now for the inducers, here are some examples of 3 strong inducers. So when a drug induces the CYP enzymes, it increases the production and activity of the enzymes, which will result in an increase in the drug metabolism for that specific drug that binds to the enzyme. Let's take a look below. Drug A is a CYP enzyme inducer. When you take this drug, it will act on the CYP enzymes, and specifically in this case, these are the CYP enzymes that normally break down drug B. And this will lead to an increase in enzyme production and activity, and more drug B will be metabolized. Next, we have the inhibitors. And here are 3 common strong inhibitors. So opposite of inducers, the inhibitors decrease the activity and production of the CYP enzymes, which will result in a decrease in drug metabolism. So once again, you have a drug that is a CYP enzyme inhibitor. So after taking it, it will reduce the production and activity of the enzymes, which will then lead to a decrease in drug metabolism. Okay, so let's take a look at some examples. By the way, if you're enjoying this video, please make sure to hit the like button. So drug A is metabolized by the CYP3A4 enzyme. And we have a patient who's on drug A and loves to drink grapefruit juice. So the mechanism is simple, right? Grapefruit juice is an inhibitor of CYP3A4. So in this case, it will decrease the metabolism of drug A and increase its levels. So based off this, what can we do about it? Now in clinical practice, depending on what drug it is, an increase in the levels can be detrimental. So we would completely avoid this interaction. So it will be a contraindication. Now for some drugs, you can reduce the dose and you can possibly counsel the patient to avoid grapefruit juice. So drug B is metabolized by CYP1A2. We have a patient who just purchased St. John's worts over the counter. So in this case, the mechanism is that the patient is on drug B and the St. John's worts is a strong inducer. So in this case, it will increase the metabolism of drug B, which will lead to a decrease in its levels. Once again, depending on the drug interaction with that specific drug, depending on what drug that is, you may want to avoid the combination, right? So it will be a contraindication. You can counsel the patient once again to avoid taking that medication, the St. John's wort, and then also you can increase the dose of drug B as needed. So before I end this video, I wanted to apply what we learned to prodrugs. Now, prodrugs are simply drugs that need to be activated first in order to actually work. And the CYP enzymes are sometimes the catalyst for this. When dealing with drug interactions, so if we have a patient on a prodrug and a CYP inducer, this will lead to more enzymes that will convert this prodrug into the active form. So same scenario, but the patient is on an inhibitor of the CYP enzyme. So this will lead to less enzymes that will convert the prodrug to its active form. And that will be all, folks. As always, make sure to show some love if you actually enjoy.
So then let's jump into his PGP explanation.
Guess which student has a better chance of getting
And their role in drug interactions. I've already put out a similar video for the CYP enzymes, so make sure to check it out. I'll put the link down below and at the end of this video. Now you see that like and subscribe button? Please hit that button. Remember, it costs $0 to do this, so please just show some love. So this is kind of how the P-glycoproteins look. It's known as an efflux drug transporter. So its job is to kick the drugs from inside the cell to the outside. Now these proteins can be found in different parts of your body. It may line the blood vessels and kick drugs out into the blood, or in the colon where it will kick the drugs back into the colon to be excreted. Same for the kidneys, the liver, and it helps protect the brain from any foreign substances also. Now because of all that I just mentioned, these proteins can affect absorption, distribution, and elimination. Now the downside to these P-glycoproteins sometimes is it's able to kick drugs out and it can lead to drug resistance. So this could be found in tumors and also microorganisms such as bacteria and fungi. But wait, there's more. We have drugs that actually induce these proteins. So in this case, we have drug B, which is a P-glycoprotein inducer. So it will induce a P-glycoprotein that normally drug A is a substrate of. Well, now not only do we have more of these proteins, but they're also working pretty well now. So it will end up kicking a lot of drug out the cell, leading to a reduction in the distribution, the therapeutic effects, and increasing the amount that's possibly eliminated. Very similar to the CYP enzymes, this interaction can be strong or moderate. And here are some examples of some strong, moderate P-glycoprotein inducers. So the inhibitors of the P-glycoprotein lead to an opposite effect. Now, we have drug B, which will inhibit a P-glycoprotein that drug A is normally a substrate for. Let's see what happens. So because of the inhibition, the drugs that are substrates of the P-glycoprotein inside the cell won't get kicked out, leading to a potential increase in the therapeutic effect. And here are some examples of strong to moderate inhibitors. And that would be the end of this video, very straightforward. And I hope I was also able to
illustrations that probably explain it better than I was doing. I think those can be very helpful. Remember that some drugs can inhibit or activate both the CYP450 system and the P-glycoproteins at the same time, specifically grapefruit juice, which is why almost always it's like if you have a question on a test and the grapefruit juice isn't a possible answer of things that can interact with the drug, it might very well be grapefruit juice. So if we know about the patient's pharmacogenomics specific to them, we can decrease adverse drug reactions. The CYP450 enzymes in families 1 to 3 mediate up to 80% of phase 1 metabolism of drugs. We know that variants in the VKO-RC1 and the CYP2C9 can affect how people respond to warfarin as well. So, implications for practice. If someone is taking 4 or more drugs, consider pharmacogenetic testing. If they're not responding to the drug as expected, consider pharmacogenetic testing. Age over 65, over 40 with chronic diseases. If you have medications that have a very narrow therapeutic window, or you'd have severe consequences of failure, consider pharmacogenetic testing. If you have a family history of serious adverse drug reactions like malignant hypothermia, pharmacogenetic testing. Or if you have someone who's adopted and they absolutely have no idea about their health history, you could consider that as well. The FDA will require tests for specific drugs like cetuximab and trastuzumab. You have to test for EGFR and HERS2 or NEU overexpression. The FDA recommends testing for specific drugs, especially for warfarin or azathioprine, and sometimes you can do it for specific drugs, but the FDA information only for the specific drugs is noted here for tamoxifen and fluoxetine. The table 6.6-6 in your book is a good example there. The other good example is that in December of 2007, the FDA added a boxed warning on carbamazepine label recommending testing for the HLA-B allele in patients with Asian ancestry before they started. So you may see some of this language in practice, and now we can better understand what it means. The future of pharmacogenetics, the costs are finally decreasing. Before it was just plain flat out cost prohibitive and insurances weren't covering it. We are seeing now that insurance is paying more and more for it. It actually can save them money because if you can get this test and more directly give them give the prescriber the right information to give them the right drug, you're going to have less adverse drug reactions, less attempts at multiple drugs. It's going to cost less if you do it better the first time. So pharmacogenetics will help, genomics will help identify the correct therapy for patients. Genotyping may be helpful to personalize the drug treatment and increase efficacy, and pharmacogenomics has significant clinical implications. Remember that sending someone for pharmacogenomics will help you really understand if they're CYP producers or users, etc. So you'll test for a plethora of drugs and you pretty much get back a report that says they can metabolize this drug, or they would be poor metabolizers potentially of this drug. So it's helpful if you send somebody like for trying to figure out the right antidepressant or antipsychotic medication. You send them for the testing, you're going to get a list of a plethora of different drug options. So you'll get SSRIs 1, 2, and 3. They would metabolize this appropriately. But SSRIs may be 5, 6, and 7. They may be poor metabolizers of. Okay. That's very helpful, but it doesn't tell you that give Sarah drug A, she will tolerate it well, she'll feel good on it, and that's the right drug to use. No. It just would tell you that I can metabolize that drug. So I think it's important to set expectations appropriately with patients and help them understand. I still may have some trial and error when I'm trialing these drugs for you. I just will know now which ones you can metabolize well and the ones you don't. So it helps me eliminate ones that I know off the bat won't work well for you, but it doesn't tell me which one's the best for you. And that's a really important point to make sure we drive home to the patients. All right, our knowledge checks time. Which of the following contributes to the variation in warfarin? So that's our CYP2C9. Which of the following are patient situations where pharmacogenomic testing is recommended? And that would be A. If they're taking 4 or more drugs or they're not responding the way we expect them to. That was your quick and easy chapter 6. These chapters are slow and sweet, which is lovely, but they do get more challenging as we move on out of this kind of first section. But for right now, we're rolling. We'll see you in chapter 7.